Bismillahirrahmanirrahim. Assalamualaikum and salam sejahtera. My name is Supriyani binti Mustamin. I am a biology lecturer from MiCoST. Okay, so today we will doing a test about blood grouping test. Okay, so before we continue for this test, I will explain about the principle behind this test. Okay, so ABO, blood grouping. Okay, so blood grouping test is a purpose for this for doing this test is to know what type of blood that we have, either A, B, AB, or O. Okay, so in our blood, okay, we have component component cell. Okay, so in our blood, okay, we have cells which is RBC, red blood cell, okay, red blood cell, white blood cell, and platelet. Okay, so in this test we will specific for RBC, red blood cell. Okay, so to know what type blood that we have, A, B, AB, or O, so we specific. So today we will test for red blood cell only to know what type blood that we have. Okay, so before that, okay, we know this is our RBC. Okay, our RBC is biconcave shape. So on surface, our RBC, they have an antigen. Okay, antigen. So this antigen is used to know what type blood, okay, that we have. Okay, so example of if we have blood type A, so on surface of our RBC they have antigen A. Okay, antigen A. Okay, antigen. And if we have blood type B, we have antigen B. Okay. And for rhesus, okay, we know we have rhesus positive and rhesus negative. So for this rhesus, they also have antigen. Okay, for this rhesus. So mainly antigen D. Okay, antigen D is to detect we have rhesus positive. Okay, rhesus positive. So example, if I am blood type A rhesus positive. So it means that in our surface RBC we have antigen A and rhesus D. Okay, antigen D. So this means we if on surface our RBC, okay, they have antigen A and D. This meaning that our blood type is A rhesus positive. Okay, so how to detect this antigen? So we use antibody to detect the presence of antigen antigen on our RBC. So to detect antigen A, we will use anti-A antibody. Okay, I will use short form for antibody. So anti-A antibody will detect the presence of antigen A. And for antigen B, so anti-B, okay, antibody will detect the presence of antigen B on our RBC. So for antigen D, okay, antigen D, also same. So antigen D will be detected by anti-D antibody. Okay? So we know we see this this antibody will specific, okay, detect their own antigen. Okay, so anti-A antibody will detect the presence of antigen on surface RBC. Anti-B antibody will detect the presence of antigen B, and anti-D antibody will detect the presence of antigen D on surface RBC. Okay, so after the detection of anti-A antibody detect the presence of antigen A, so example for structure anti-A antibody. So this is the structure of antibody, okay, Y shape, and at the Y shape there is one site called antigen binding site. Okay, antigen binding site. Okay, so example, this is the structure for anti-A antibody. Okay, anti-A antibody, its shape is Y structure, and there are site for antigen binding. We call this antigen binding site. Okay, and the antigen binding site is the shape is specific triangular shape. So the antigen binding site for this anti-A antibody is also triangle shape. Okay, so it means that antibody, okay, will recognize the antigen specific. Okay, specific. So if this anti-A antibody bind to antigen A, okay, for example, after the binding for anti-A antibody to antigen A, okay, they will one response is called agglutination. Okay, agglutination. Okay, atau kita panggil dalam bahasa Melayu gumpalan. Okay, so when we doing this test, we will see the reaction agglutination is our RBC will agglutinate. Okay, agglutinate in the blood. Okay, it means that antibody for this antigen is bind to the antigen. Okay, so anti-B antibody will bind to antigen B, and anti-D antibody will bind to antigen D. So when the binding occur, agglutination will occur. Okay, so this is the principle, okay, behind this test. Okay, so if this agglutination occur, it means that the antibody detect the presence of antigen. Okay, so to proceed our blood grouping test, okay, I will assist by Encik Zul, okay, our lab assistant. Okay, so thank you Encik Zul. Okay, so before proceed, I will explain. So this is the material. Okay, this is the material that we'll use for this test. Okay, so we need lancet, okay, to take blood. Encik Zul blood, okay, I will use Encik Zul blood as a sample. Alcohol pad, okay, spot plate to put antisera and blood in this, okay, and toothpick, glass pasture pipette, okay, glass pasture pipette and antisera. Okay, so this is the main important thing in this test. Okay, so what the content of this inside antisera? So in this antisera, we see there is colorful. Okay, so for blue color, okay, antisera. So there is antibody, anti-A antibody in this serum. Okay, serum. Okay, so A means there is presence of anti-A antibody in this serum. So what the use of this anti-A antibody? To detect the presence of antigen A, okay, on surface RBC. Okay, so this yellow antisera contain anti-B antibody. okay, to detect the presence of antigen B on surface of our RBC. So this clear reagent contain anti-D antibody to detect the presence of antigen D on surface RBC. So anti-D, the function is to know either we rhesus positive or rhesus negative. Okay. Okay. So first of all, we put anti-sera, okay, on spot plate. Okay. Before that, we label this. Okay, so we put antibody on plate label A, 1 drop only. Okay, then I will take a sample blood from Encik Zul. May I? Yes, thank you. Okay, so make sure you swab. Okay, so what the use of this alcohol swab is to clean the skin, the finger, before we take a blood. This is to avoid from infection. Okay, so this is used to kill bacteria, okay, on the skin. Okay, make sure just swab one way sahaja. Okay. Boleh. Okay, so this is lancet to prick the finger. Kenapa tu? Oh, dah. Sekejap. Okay, Zul. Sebelum tu, Zul. Nak lagi ke, Zul? Okay, dah cukup. Bersih kan ni? Macam kotor je. Okay, just drop one drop of blood. Okay, make sure the not mixture. Okay, then we use toothpicks to mixture the blood and the anti-sera. Okay, make sure don't mix the toothpicks. Okay, and we leave it for 2 minutes. Okay, a few minutes to see either they are agglutination or not. Okay, so now we see the reaction for anti-sera, antibody with Zul blood. Okay, so there is agglutination in A, meaning that antibody contained in this anti-sera detect the presence of antigen A on surface of Zul's RBC. Okay. On B also there are agglutination, and D also agglutination. This is meaning that Zul is AB, blood AB rhesus positive. Okay, so this is another blood sample from mine. Okay, so we see there is no agglutination occur on AB. Okay, look the differences with the above sample, but there is agglutination on D. So this is meaning I am blood O rhesus positive. Okay, because there is agglutination or gumpalan on D. Okay, that's all. Okay, thank you, Zul. So that's all for today from us. Please like and subscribe and share. Please like, share, and subscribe. Okay, thank you very much. Assalamualaikum, bye.